Assalamu alaikum wa rahmatullah. This is the immunology chapter. Starting with functions of the immune system. Defense against organisms. Okay, very important. Recognition of self and non-self antigens. And this regulation of this function will end by autoimmune disorders. Also, recognition of oncogenic stresses and elimination of malignant clones. So, the immune system has three functions: to defend against organisms and to recognize self and non-self, and so to avoid attacking the non-self antigens, and defense against malignant transformation. Components of the immune system. This includes the lymphoid organs and immune cells. The lymphoid organs, we have central lymphoid organs, including bone marrow and thymus, that produce the lymphocytes. And we have peripheral lymphoid organs, where naive lymphocytes, which are immature or non-specific lymphocytes, meet antigens, okay, and presenting to them by antigen-presenting cells. And then they initiate an adaptive immune response. This meeting between the naive lymphocytes and the antigens occur in lymph nodes, spleen, mucosal-associated lymphoid tissues present in the gut and airways. So we have central and peripheral lymphoid organs, central that produce lymphocytes, and peripheral where lymphocytes meet antigens to induce a specific immune response. Then the second component of the immune system is cells of immune system. Cells of immune system start with the multi-potential hematopoietic stem cell, okay, in the bone marrow, that then can be differentiated into common myeloid progenitor and common lymphoid progenitor. The lymphoid progenitor produce natural killer cells. This is one of the innate immunity cells that share in both innate and adaptive immunity. And the second is the lymphocyte. Okay, so common lymphoid progenitor gives us natural killer cells and lymphocytes. And lymphocytes will be differentiated later on to T lymphocytes and B lymphocytes that differentiate into plasma cells producing antibodies. The other side has a common myeloid progenitor, and this line that will be differentiated into megakaryocytes that give us platelets, erythrocyte that gives us red blood cells, and then the remaining white blood cells include mast cells and myeloblast. Myeloblast will differentiate into basophil, neutrophils, eosinophils, and monocytes. Okay. Monocytes circulate in the blood, okay, or present in the circulation, and when it resides in the tissues, it's called macrophages. So we have granulocytes that include all these. Okay, include basophil, neutrophils, eosinophils, mast cell. All these are granulocytes, and the agranulocytes that includes monocytes and lymphocytes. We have stem cell, okay, followed by common myeloid progenitor and lymphoid progenitor. The lymphoid progenitor produce natural killer cells and lymphocytes, while the myeloid progenitor produce the erythrocytes, megakaryocytes, and the granulocytes, including basophils, neutrophils, eosinophils, and agranulocytes, including monocytes that differentiate into macrophages in tissues. So these are granulocytes, okay, and these are agranulocytes, lymphocytes, and monocytes. All these constitute the white blood cells. Okay, so we have first line immunity against stresses confronting the body. It's called the innate immunity, and the second line, which is the adaptive immunity. We have one cell that has role in both adaptive and innate immunity, called natural killer cells. Adaptive or specific immunity associated with a memory, usually the role of B and T lymphocytes. T lymphocyte constituting cellular immunity, and the B lymphocytes which secrete or produce antibodies constituting a part of the humoral immunity. The innate immunity, we have natural killer cells again, we have the monocytes, macrophages, we have neutrophils, mast cell, eosinophils, basophils, and dendritic cells. Starting with the first line defense that initiate adaptive immune response later on, which is the innate immunity. Components of innate immunity including direct barriers, normal barriers, which can be physical like skin and the mucous membranes, for example, can be chemical barrier and can be biological barrier. So direct barrier including the gastric HCl, for example, vaginal secretions, alkaline and acidic pH at different areas of the body, bile and pancreatic juice, and so on. So direct natural barriers is part of the innate immunity. Then we have cellular immunity, cellular component formed by granulocytes and agranulocytes as a part of the innate immunity. And we have humoral part, humoral mechanisms that include the complement system circulating in the plasma, chemokines, and interferons. So these are the components of innate immunity. The first line defense that will initiate adaptive immune response on re-exposure. These include direct barriers, cellular mechanisms performed by granulocytes and agranulocytes, and humoral mechanisms composed of complement system, chemokines, and interferons. So functions of innate immunity include recognition, by certain receptors on the surface of macrophages, neutrophils, dendritic cells. These are called toll-like receptors. These receptors can recognize any foreign pathogen or material invading the body. Also, they have function of acute inflammatory response. Once finding this foreign material, so phagocytosis and intracellular killing by macrophages and the neutrophils is very important. So inducing acute inflammatory response is the responsibility of innate immunity. Secretion of cytokines and the chemokines by macrophages and the neutrophils that further augment the inflammatory response initiated initially by recruiting more effector cells, which mainly lymphocytes, to the site of infection. So they secrete cytokines and the chemokines to augment the inflammatory response and to trigger the adaptive response. Induction of adaptive immune response by activated antigen-presenting cells. Antigen-presenting cells, like dendritic cells, migrate to the lymphoid organs we have mentioned as lymph nodes, spleen, and so on. So where they present the foreign antigens to the T and the B cells to initiate the adaptive immune response. These are functions of innate immunity. Cells of the innate system includes the phagocytes. This is the process of phagocytosis and intracellular killing of a foreign material. So phagocytes including neutrophils or polymorph nuclear cells, and the mononuclear cells, which are monocytes in blood and macrophages in tissue. These are the phagocytes, polymorph nuclear and the mononuclear cells. Neutrophils destroy pathogens by superoxides and proteases. This is very important, and as we have a disease called chronic granulomatous disease, has dysfunction of this phagocytic cells, or phagocytic function, so will end by immune deficiency. Macrophages present antigen to T cells. So we have phagocytes, we have dendritic cells, and we have the natural killer cells, which kill virus infected cells and the tumor cells. natural killer, very important cell in the immune system, has a vital role against virus infected cells and against oncogenesis. So neutrophils and macrophages perform their actions via phagocytosis and intracellular killing, while tissue dendritic cells are activated by pathogens and then present cells to the naive T cells in the peripheral lymphoid organs. And macrophages and dendritic cells are antigen presenting cells, activate T cells and initiate adaptive immune response. Complement system. Okay, complement system is an important component of the innate immunity, which are composed of a group of plasma protein. These plasma proteins are activated by pathogens or pathogen-bound antibody, either the antibody catching the antigen or the antigen itself will activate the complement system. This system constitutes a link between innate and adaptive immunity. There are three pathways discovered that activate the system, resulting finally in opsonization, chemotaxis, cell lysis, or inadvertently anaphylaxis. Okay, so stimulation of a complement system will end by the immune response in the form of opsonization, chemotaxis, or cell lysis. Okay, and in some cases that are abnormal, this can be exaggerated, ending by anaphylaxis. So starting with the classical pathway, classical complement pathway is called this because it was the first one to be discovered, where binding of antigen to antibody leads to activation of the C1 complex. Okay, C1 complex that includes C1q, C1r, and C1s. So we have three pathways for activation of the complement system that all ends by activation of the C3. Okay, C3, this is the component, one of plasma protein, one of the component of the complement system. We can call it a common pathway for the complement system. So these three pathways for activation of C3 include classical pathway, the first one to be discovered, and the alternative pathway, the second one to be discovered, so it's called alternative pathway, and the lectin pathway, which is the last one to be discovered. So this is the classical pathway where having a microbe that invade the body has antigens on its surface. Okay, where it will be recognized by antibodies. Okay, antibodies catching the antigen, forming the antigen-antibody complex. This activates the C1 complex. When it starts with the C1 complex, it's called the classical pathway. Activation of C1, okay, with its component C1q, C1r, and C1s will result later on by activation of C2, C4, and ending finally by activation of C3. Activation of C3 gives us the C3a, C3b, okay, that will end by the immune response in the form of degranulation of mast cell, release of histamine and other inflammatory mediators, opsonization of the microbe or the antigen, and cytolysis, okay, destruction of the cell of the microbe or the infected cell. This is the classical pathway. The alternative pathway, okay, the second one to be discovered, so having a microbe with lipid carb complexes on it will activate certain complement system components. These are called B factor, D factor, and P factor, which are called properdine, and this activation will end by the C3 activation. And again, C3 activation will end by the immune response in the form of degranulation of mast cells, release of inflammatory mediators, cytolysis, and opsonization. Okay, this was the alternative pathway. Finally, the lectin pathway, last one to be discovered when a microbe invading the body, which activate liver to secrete mannose binding lectin. This is one of the acute phase reactant. Okay, having a microbe with these carb containing mannose on its surface, like certain organisms has this carbs on its surface, will result in activation of the lectin pathway, and then activation of lectin pathway will end also by activation of C3 and the same immune response. So complement system is a vital component of the innate immunity. It's a humoral component, plasma protein circulating in the blood, have 3 pathways for its activation, which all end by activation of the C3, very important. We have classical pathway, first one to be discovered, and then alternative pathway, and then lectin pathway. Activation of these will end by activation of C3, giving us the characteristic immune response. So the 3 all generate C3 convertase, cleaves C3 into C3A and C3B. Subsequent effector mechanisms include opsonization, chemotaxis, anaphylatoxin, which will end by mast cell degranulation, and formation of the membrane attack complex. Membrane attack complex is formed by C5, C6, C7, C8, C9. So from C5 to C9, plasma protein, complement plasma protein. This is called membrane attack complex that is involved in the cell lysis. Okay, these are the effector mechanisms of the complement system that can start in different ways in the 3 pathways depending on the stress inducing the response, and all end by a common pathway, which is the C3 convertase that gives us the active C3 that will end by these effector mechanisms: opsonization, chemotaxis, mast cell degranulation, and formation of the membrane attack complex that will end by cell lysis. It's very important to have down regulators for the complement system, otherwise spontaneous activation of the complement system can give us disease. Okay, for example, if you remember in the nephrology chapter, we have a typical hemolytic uremic syndrome, okay, and the thrombotic thrombocytopenic purpura. These disorders have continuous activation of the complement system, resulting in a disease. So and this usually caused by deficiency of these down regulators of the complement activity, which include factor H, factor I, and the C1 inhibitor. These shut down the proteolytic activity of C1 component of the complement system. Also having membrane control protein like decay accelerating factor. Having also decay accelerating factor on the cells is very important to down-regulate the complement system, otherwise disease will result. If you know the paroxysmal nocturnal hemoglobinuria, it's one disorder associated with hemolysis and the nighttime hemoglobinuria. It's caused by deficiency of this important down-regulator of the complement system, which is the decay accelerating factors. So we have mentioned now the 3 pathways. What is the complement system? The 3 pathways for its activation, the common pathway, its effector mechanisms or the immune response, and the down regulator of the system, very important, mentioning 2 disorders that can result from deficiency of these down regulators, which usually result from genetic mutation. Now we are in the humoral mechanisms of the innate immunity. After complement system, we have cytokines and the chemokines. Cytokines, these are soluble factors that link immune system produced in response to pathogen recognition. This includes interleukins, interferons, and tumor necrosis factor alpha. So exposure to one pathogen will result in activation of the innate immune cells, and the innate immune cells, including neutrophils, macrophages, will secrete cytokines that mediate the inflammatory response, including interleukins, interferons, and the tumor necrosis factor. Chemokines, these are chemoattractants that recruit more and more neutrophils, monocytes, and other effector cells to the site of the infection. Interferons and cytokines secreted by macrophages activate natural killer cells, which control intracellular infections, especially viruses. Okay, we have mentioned natural killer cell has a very important role against virus-infected cells and the tumor cells. Activated by these interferons and the cytokines secreted by the macrophages. Cytokines and the chemokines are also secreted by lymphocytes of the adaptive immune system. So this was the innate immunity. Now, talking about the adaptive immunity. Adaptive immunity have both humoral and cellular components. Mainly lymphocytes that initiate the adaptive immunity. They respond selectively to different antigens. This is the difference between innate and adaptive immunity. Here we have a selective immune response. Okay, we target certain antigen by a specific lymphocyte. So development of defense mechanisms, okay, by adaptive immunity, very important, and adaptive immunity and by formation of a memory, okay, of a specific exposure, and this constitutes the basis of immunization, to be exposed to certain antigen, and that will be followed by formation of a selective or a specific adaptive immune response. On re-exposure, we have memory cells that will initiate a selective immune response. The humoral component is formed by P lymphocytes through production of specific antibodies, and a cellular component which is T cell dependent, okay, that will result in macrophage activation and cytotoxic T cells. So the first function again will be recognition, okay, by the adaptive immunity, the selective immunity, the specific immunity. We need to recognize the invading pathogen or foreign antigen. This is function of antibodies. They have recognition receptors. Also T cell receptor, TCR or T cell receptor, very important. Recognize antigen that are bound to the MHC molecules or the major histocompatibility molecules. These molecules present antigens, different foreign antigens, to T cell receptor or the TCR. These we have two classes of the major histocompatibility complexes. Class 1 found in all cells of the body, okay, and class 2 present on P lymphocytes, macrophages, and dendritic cells. Okay, these antigens bound to foreign bodies and present them, so they recognize the foreign antigen and present them to the effector T lymphocytes. Okay, also we have natural killer cell receptors. So recognition of the foreign material to initiate the adaptive immunity is a function of antibodies, T cell receptors, okay, and natural killer cells. Lymphocytes. Starting with the T lymphocytes. T lymphocytes, they are thymus dependent, as they originate initially in the bone marrow, and then they need to maturate in the thymus. Okay, where the TCR or the T cell receptors, that's very important for recognition of the foreign material, is formed or combined to the T lymphocyte in the thymus. Okay, so maturate in the thymus through formation of TCR to the naive CD4 and the CD8 T cells formed in the bone marrow. Okay, then become activated in peripheral lymphoid organs after exposure to different antigens. After they are activated, clonal expansion of lymphocytes and further differentiation later into the effector T cells occur upon exposure to antigen presenting cell. Okay, so we start in the bone marrow forming naive T lymphocytes that migrate to the thymus to combine the T cell receptor or TCR, then in peripheral lymphoid organs, including lymph nodes, spleen, lymphoid tissues in the gut or the airways, they meet the antigen presenting cell with their code antigens, and this will result in their differentiation into effector T cells that will perform the functions of the adaptive immune system. Some of these T cells and B cells differentiate into memory cells, very important, which promptly differentiate into effector cells upon re-exposure. Okay, so all these T lymphocytes express the T cell receptor, TCR, very important, and further mature into the effector T cell. What are the effector T cells? We have the helper T cells or CD4, okay, and by CD4 cluster differentiation, we cluster of differentiation. We have certain antigens here on the surface of all cells, and this can be recognized by the flow cytometry and given numbers. Okay, there are more than 300 numbers of cluster of differentiation are available for recognition of the cells. Okay, so T helper cells are given the number cluster of differentiation 4, and the T cytotoxic cell are giving the number CD8, and so on. So effector T cells after maturation include the T helper cells, and these are essential for most antibody and the cell mediated responses. T helper cells divide into T helper 1 and T helper 2 and T helper 17. They have different functions, okay, either activation of the B cells or having a role in the autoimmune disease, or activation of the macrophages and CD8. Okay, so T helper cells or CD4 activate CD8, activate macrophage, activate B cells to produce antibodies, okay, and have a role in autoimmune disease. Then the T cell activate the T cytotoxic cell or CD8. This is the cytotoxic T cell or CD8 activated by the T helper cell. This has a very important function in killing cells, expressing their specific antigen target. Okay, and then we have regulatory cells having the cluster differentiation 25. And this inhibit target T cells or release inhibitory cytokines interleukin 10. So it has a regulatory function. Otherwise, uncontrolled immune response may be harmful to the patient. So again, these T cells all have a T cell receptor on their surface for recognition of antigens by the antigen presenting cells, and then further mature into the effector cells. These effector cells include CD4 and CD8 and the T regulatory cells. CD4 is very essential for different functions of the adaptive immune system, including T helper 1, 2, and 17, activating macrophages, T cytotoxic cells, and B cells. The T cytotoxic cells very important in cellular killing, okay, very important, and the regulatory T cells very important for regulating the T cell function, guarding against uncontrolled immune response. They secrete the interleukin 10. B lymphocytes also originate in bone marrow through different stages, from pro-B to pre-B to mature B cells. These B cells migrate to peripheral lymphoid organs, lymph nodes, gut-associated lymphoid tissue in the airway, spleen, where they are activated by antigens leading to proliferation, differentiation into plasma cell, that is antibody-producing cells. Antibody, these B cells use the antibody as their antigen-specific receptor. So we have here a plasma cell that is a mature B lymphocyte which secrete the immunoglobulins or antibody, and this is the immunoglobulin or the antibody. On encountering antigen, differentiate into plasma cell and secrete antibody. What about this immunoglobulin molecule or antibody? So it constitutes a cell surface receptor on the B lymphocyte. If this is the B lymphocyte or plasma cell, it has on its surface the antibody as a antigen specific receptor which will recognize the foreign antigen. It has two heavy and two light chains. These are light chains and these are light and these are heavy chains, giving a Y shape. Each heavy or light chain is composed of constant and variable regions. These are the variable region or Fab fragment, and this is the constant region or the FC fragment. The constant region defines effector functions of the antibody, while the variable region corresponds to the antigen binding site. Okay, so again, this is the Fab fragment or the variable region of the immunoglobulin that recognize the different antigens. Okay, while this is the FSC portion which determines the effector functions of the antibody. FEC region has complement activating domains also, which will fix the complement system. Effector cells recognize antibody-coated pathogen by their FEC receptors, which bind to FEC region of the antibody. So, effector T cells, okay, has or have receptors for the FEC portion of the antibody on their surface for recognition. The synthesis of the immunoglobulin has stages, stage 1 recombination and stage 2 class switching. The VDJ recombination as a variable region of the antibody is encoded by genes segments called variable diversity and joining segments. Okay, so recombination of gene segment to produce the variable region. This is the first stage of antibody synthesis. Then class switching is a very important stage, as initially only we have IgM and IgD produced. So the first immunoglobulin produced is the IgM, okay, and then switching from IgM to other different immunoglobulins occur, okay, to IgA, IgE, IgG, and IgD immunoglobulins. So we have important entity called the hyper IgM syndrome that is X-linked condition where dysfunction occurs here at this stage, resulting in failure of class switching, which will end by very high levels of IgM molecules and low levels of other Ig molecules. We have 5 classes of immunoglobulins which are based on the heavy chain type. Okay, we have the IgM, IgG, IgA, IgE, and IgD, which and then IgG is further divided into subclasses from IgG1 to IgG4. This is the antigen binding site or the Fab fragment or the variable region of the immunoglobulin that contain both light and heavy chains, and these are the FEC portion, okay, formed of heavy chains that determine the effector function of the antibody and that can affect complement system, okay, and has and constitutes the antigen specific receptor on the B lymphocyte. Also has a receptor on the effector T cells for its binding. So these are the criteria, different features of different immunoglobulins, with the immunoglobulin G is the only one that can cross the placenta, okay, giving immunity to the fetus that will continue with the baby till about 6 months of life. okay, protecting him against different pathogens in the early life, while the IgM molecule constitutes the largest one that cannot cross the placenta. This is very important. Generation of immunological memory, as we have mentioned, specific immunity mediated by humoral and the cellular system. This immunity is long-lived antigen-specific lymphocytes, okay, induced by natural infection or by either immunization or natural infection, can result in formation of long-lived antigen-specific lymphocytes. Okay, on re-exposure, these lymphocytes will, these are called the memory cells that will recognize the foreign material and initiate the immune response. Memory is learned during the development of the immune system, which explains the difference in frequency of infections between adult and children.